So my project was on tracing ancient and modern circular DNA elements across millennia. So what that means is for my project, I compared ancient and modern samples. So two types of circular DNA, plasmids and crest DNA viruses. Plasmids are small circular pieces of DNA found in bacteria. And they're important because they often carry antibiotic resistance genes, which help bacteria survive treatments with antibiotics. Crest DNA viruses are another type of circular DNA element. They have special proteins called replication associated proteins, which are needed for them to make copies of themselves and are used to classify themselves. So I studied both because plasmids and crest DNA viruses are mobile genetic elements. So that means they can move between organisms and affect different communities of organisms. So by comparing these ancient and modern samples, we can see how genetic elements have changed or stayed the same over thousands of years. And this has been really important because it helps us predict future risks, like how antibiotic resistance might spread or how new viruses could appear. So in this, these are both two pictures. On the right, you have plasmid DNA in a bacteria, and on the left, you have a genetic tree of different crest DNA viruses, because crest DNA viruses is a very broad term to describe DNA viruses. So why would we compare ancient and modern samples? So in our study, we compared DNA from an ancient peat bog, which is estimated to be roughly 11,000 years old, and modern samples from Mill Creek in Arizona. So the main reason for doing this comparison is to see which genetic elements, like plasmids and crest DNA viruses, have persisted over time and how they've changed or adapted by tracking evolutionary changes. So we can learn how microbial communities respond to environmental pressures over time with this research. And how this matters for public health, which is another lens I viewed my project through, is because these mobile genetic elements can still shape microbial communities today, even though they were from 11,000 years ago. So for example, like I said, plasmids often carry antibiotic resistance genes. So if certain genes or replication strategies have survived over those thousands of years, it suggests that they are very stable and could continue to influence bacteria in the future. So that helps us predict whether resistance traits will stick around or evolve in new ways. So I chose this topic because I first learned about antibiotic resistance in the 9th grade. It's when I watched a documentary called Fighting the Nightmare Bacteria. And this documentary really stuck with me because it showed how something this microscopic could create such a big global health crisis, because there is a global health crisis going on right now with antibiotic resistance. So this curiosity led me to research more about antibiotic resistance. So this project just felt like the perfect mix of learning about evolution while also connecting it to things like public health. And one day I hope to go into medicine and study public health. So this is an exciting topic to explore. So for my research question, I set out to understand how plasmids and viruses from an 11,000-year-old peat bog compare to those from a modern creek. And I was asking the question, what does that tell us about their evolution and the public health implications of that? So I basically wanted to find out which genetic elements have persisted and which have changed over thousands of years. So as an overview of my methods, we started by extracting DNA from the ancient peat bog from soil, and then we assembled the sequences. We also did this for the modern creek sediments. Using PROCA, which is a data analysis tool, we annotated these sequences to identify genes and open reading frames. So we focused specifically on plasmid and viral elements. So for the viruses, we tried to look for replication-associated proteins and capsid proteins, which are like key markers of a CREST DNA virus. And then for the plasmids, we were looking for antibiotic resistance genes. So after identifying these genes, we sequenced them using PSI-Copy, which is an alignment tool, to compare their evolutionary differences. And then we constructed an phylogenetic tree using PhyML to visualize the relationships between these ancient and modern sequences. So when we started with gene annotation and antibiotic resistance screening. So by annotating all the sequences using PROCA, we were able to have like functional predictions for both the ancient and the modern samples. So the key results with this is that the replication-associated proteins were very abundant in our viruses, but completely absent in our plasmids. So in total, we found 4 replication genes in the ancient peat bog and 49 reps in the modern creek viral sample. So in contrast, the plasmids had no rep proteins, like I said, and they also had no antibiotic resistance genes. And then we were confirming our screening with a CARD database, which is an antibiotic resistance database. So our results suggest 2 things, which is first that the viruses were functionally active in both the ancient and modern environments, and that second, plasmids did not act as reservoirs for resistance. It's at least in our sample, they did not. So on the left, this is an ancient plasmid genome map, which is about 4,000 base pairs long. It contains 8 predicted coding sequences, but all were annotated as hypothetical protein, so they were unable to identify any proteins that in this database, probably because they are so ancient that there is nothing to compare them to. And then on the right, we have an ancient viral genome. This one is about 4,000 base pairs long as well, and it contains a replication-associated protein and a capsid gene. So these are 2 of the very these are 2 big indicators of CREST viruses, and this is confirming its viral identity, and it highlights the major differences between plasmids and viruses in our data set and how we were able to classify the viruses, but not the plasmids. So next we did our phylogenetic tree analysis. So our tree includes 53 replication-associated proteins from both ancient and modern samples. We built this using the PSI-Copy alignment and PhyML. So the main takeaway is that the ancient rep in the phylogenetic tree did not sit alone. It clusters with a modern clade that suggests there is some continuity from the ancient and the modern DNA sequences. And the important clarification is not all of these sequences belong to the CREST DNA group. In the red box, that is all phages, which are viruses that infect bacteria. This was a very interesting find. They still encode rep proteins and share some characteristics with CREST viruses. And since phage technology is an emerging way of combating antibiotic resistance, looking at ancient phages is a really interesting way to see how they evolved as well. So for some of our results, some evolutionary insights with this would be how our phylogenetic analysis shows some viral replication proteins that have stayed almost the same for thousands of years. And this suggests that they are essential for the virus's survival, and that changing them too much would likely stop the virus from working. On the other hand, some reps have evolved and changed and have probably had to adapt to new hosts or environments. And studying this ancient DNA is giving us like a rare look at how these genes have evolved over time. However, our study has had a lot of limitations since we only had a few ancient samples that we were able to get, and that current reference databases are unable to detect some of the genes because they are so ancient. They're just not in the reference database. Some implications for public health is that one, we didn't find any antibiotic resistant genes in our plasmid samples. So, and that goes for both the ancient and modern samples. So for the ancient plasmids, this makes sense because they have existed long before humans started widely using antibiotics. So antibiotic resistance was not as common back then, but this is giving us like a baseline for understanding what these like plasmids look like before human influence. But for the modern samples, it was more surprising that we didn't find resistance genes. And that doesn't mean that they're not there. It could just mean they aren't there in that environment or the reference database just wasn't able to find them. So we'd need a bigger data set for that. Interestingly, we found phages, which are viruses that infect bacteria. And this matters for public health because phages are being explored to be used as alternatives to antibiotics. So understanding their diversity and evolution can help inform future treatments. And if I were to do more research on this, I would probably go more in depth with the phages and see how their evolutionary history looks like. For the broader impact, our study connects evolutionary biology with public health, and it compares ancient and modern DNA. So we can see which genetic elements have stayed the same over time and how they have changed over thousands of years. And this is just helping us understand how microbes adapt and spread genes. And these insights are going to help us predict future risks and develop strategies to manage antibiotic resistance and to improve phage-based therapies. Um, Again, I will reiterate that our sample size was very small and reference databases are incomplete, so more research is needed to confirm the patterns that we have seen. Okay, I'd like to give a heartfelt thanks to my mentor, Dr. Ignacio de la Higuera, for all his guidance and patience with me. And he taught me a lot of new skills. I had to learn coding for this project and a lot of data analysis. So I have to give him props for guiding me through that. And then also a huge thank you to Talaria for giving me this incredible research experience. It's been incredibly rewarding, and I'm grateful for everyone who made it possible.